And I'm your host today. My name again is Meg Inglis, and then we have Kim Conroe, who is the immediate past president, and she will be monitoring the chat function. And before we even get started, I want to say that no effort like this can ever occur without a bunch of people behind the scenes working on it. And I particularly want to thank Linda Knowles, because she's the backbone of the Spring Symposium and has been doing it for many, many years. Adam's going to be speaking to us about saving the lady slipper orchids in Texas, and I'm really excited to hear this presentation. Adam Black is a lifelong plant enthusiast with a passion for the rare, unusual, and esoteric. Based near Navasota, Texas, Adam combines his experience in the fields of botany and horticulture by promoting diverse landscapes, while also collaborating with various gardens and universities, collecting imperiled plant species for the purposes of research and conservation. He goes out into the field quite a lot with a bunch of different botanists and has a good time. I think that's how we're going to have to summarize that. Currently, he is self-employed as a botanical horticultural consultant, while also serving as a program coordinator for the Global Genome Initiative for Gardens. He previously worked at several botanic gardens, most recently as director of the John Ferry Garden, formerly known as the Peckerwood Garden in Hempstead, Texas, where he assisted with the transition of this internationally acclaimed private garden into a public garden. Adam is a member of the Pines and Prairies NIPSOT chapter and serves as their botanic advisor. So Adam, take it away, Adam. Thank you for coming.
Yeah, well, thank you all for having me. Yeah, I mean, basically, when it comes down to it, I'm just really just a total plant nerd. It's a pathological condition that basically I'm just, any waking moment I can get away from reality, I'm out chasing rare plants. But yeah, basically I'm all over the place. I'm both, a lot of what I do is for money does involve plants, but a lot of my plant work is not paid for. And we'll kind of elaborate on that a little bit into the presentation here, but I think a lot of people follow my Instagram and Facebook posts and think that I'm just getting paid to do all this, and really it's not. There's a lot of us out here who are basically just doing this too. It's not just me. It is definitely a collaborative effort among a lot of folks around the state and around the country and around the world. Actually, there's a lot of unsung heroes out there working with plant conservation. I think a lot of people think about when they think of conservation efforts, they think of the initiatives at botanical gardens and folks like Brit and other things, not their institutional conservation efforts, which they're all doing great things, but they can't do everything. And a lot of times it really comes down to a lot of these private individuals who just take the time and have the time to really get around, and as opposed to like a grant funded project where your time is limited, and maybe if you have one shot at it during 1 year, and if it doesn't work out due to weather or whatever, oftentimes those plans are kind of set back. So yeah, with the lady slipper orchids, I mean, it's one of our flashier plants of conservation concern in the state. I do work with a lot of the less flashy stuff too. I'm definitely an equal opportunity plantsman. I like all the little unsung little weedy things that also have a place in the environment as well, that other people may not even bat an eye at just because it doesn't have pretty flowers or something like that. But and I was never really into native orchids for a long time. It was just one of those things. Everybody else kind of likes them, but and I always gravitated towards the more unusual or the odd or cryptic, and not necessarily all the big showy stuff. But orchids have this kind of way of drawing you in, and so eventually I just couldn't help it, and they just became part of one of my many interests as well. I still like all the little boring things as well, or what other people would consider boring. But with lady slippers, as you can see here, it's quite diverse. There's 60 species of Cypripedium specifically worldwide. There's other species of orchids around the world that do get kind of lumped under the common name lady slipper orchid. Some of you might grow like Paphiopedilums or some of these other Asian species that have similar shaped flowers. But for the purpose of the talk, we're just going to be referring to the genus Superpedium, which is native to North America down in Mexico. But got 12 species North America. 2 are in Texas or have been recorded in Texas, but 1 may no longer exist, at least within the state lines here. We'll mention that one in a second. But Cypripedium kentuckiense is the one we're focused on. There's very few sites left in Texas. It used to be more common before either land was developed or just other changes to the hydrology or that sort of thing. And maybe other issues as well. And that's the main problem, is that each of these sites may only have 1 plant, and the possibility that those 1 plants are going to be basically self-pollinated is low to impossible. So this is where humans kind of have to intervene. So here's kind of the range of Cypripedium kentuckiense. You can see there's a number of counties that are shaded in. Basically, these are based off of county records, this map here. So just because you see a county shaded in there doesn't mean they're all over in that county. That may just be one historic record where they may not exist anymore. And some of these sites, they definitely do not exist anymore. It's just probably about half of these counties here. You can still find Cypripedium today. You can see the name kentuckiense obviously refers to Kentucky, where it was first described up there, but you can see the main portion of the population is actually further west: Oklahoma, Arkansas, Louisiana, and into East Texas here, with just those scattered records more to the east and suitable habitat. The other species we'll just touch on briefly here: Cypripedium parviflorum. The one I was mentioning is likely not present in the state anymore. There was 1 historic record way up there in the northwest Texas, in just a very harsh environment. It must have had there's very little information as far as exactly where it was collected. It was, I think, early 1900s, but it was probably just some small little microclimate where this thing just happened to be able to eke out a living, and may no longer exist there. Or it could be hidden on someone's private ranch off somewhere in some little sheltered canyon. We really don't know, but at the very least, it hasn't been seen since that original record within the state. But you can see it picks up again in New Mexico. So obviously it gets named lady slipper orchids because, well, I mean, it doesn't really look like a slipper. It's kind of inflated. I think the common name comes from some of the more European varieties of lady slippers that do actually look more like slippers rather than a big ballooned up pouch here like you see here. One thing, I was watching TV one time and it just dawned on me, it just I can't unsee this now. It just looks like Stan from American Dad with that jaw just jutting out there, but I doubt we'll ever call it the Stan orchid. But anyway, why are they rare? They're very specific in their habitats. Again, just because they're in those counties doesn't mean you can just go to those counties and just easily find them. They live in these ravines with proper geology, proper soil chemistry, and that's definitely not exposed everywhere. It's only exposed in kind of portions of maybe some of these little ravines that are cut into those particular rocks. And even with that, they've obviously been attractive to people. So people have been tempted to dig them up and try to grow them in their garden, which usually does not work out. Because again, they want those specific conditions. And if you just pull them out of that and stick them into your flower bed, they're definitely not going to make it there. Again, the remaining plants are just very widely scattered. So one plant may not be within pollination distance. In other words, a bee, these are pollinated by bees. So a bee is not going to be able to pick up pollen from one plant and be able to cross sometimes 30, 50 miles to the next closest plant that's known. And so these are basically just kind of persisting where they are. They're also seem to be dependent, this hasn't been proven yet, but they seem to be dependent on flowering dogwoods as actually like the mycorrhizal kind of partner there. They have this, as some of you may know, a lot of orchids do have, are dependent on mycorrhizae. And in this case, almost all cases, there always seems to be a dogwood nearby. So they may be connected to those, the mycorrhizae that do tend to kind of partner with the flowering dogwood. I've heard from people saying, oh no, there's no dogwoods at this site or that site. And then we go there and sure enough, there is a dogwood. It's right within the drip line. Or at some of these sites where some plants are just kind of persisting and not really flowering anymore. Whereas people do remember flowering in recent decades. It all goes back to a known dogwood that was there that's either declined or just is not present anymore. So the plants are still persisting there, but they're just in a weaker state. So it's providing some evidence that there may definitely be a connection there. Ron Dieterman from Atlanta Botanic Garden was the one that really turned me on to that fact there. On his property, he actually sprinkles Superpedium seed just around the drip line of dogwoods, and he has these rings of Superpediums that come up. But of course, he's got the proper soils on his property in Georgia too. And he's tried it around other trees, and you may get a few come up, but definitely not like it's really amazing to see what he's done both at Atlanta Botanic Garden and just on his own property. And the other thing we need to consider here is just with climate change, they may not be happy here anymore. They may want to be further north up in the Ozarks and maybe the higher elevations where it's a little bit cooler. And we just have these few plants that are just kind of just barely hanging on in these cooler microclimates in these ravines here. A lot of people just always want to blame humans for everything, and but in some cases this may be just a leftover from the ice ages too, where I mean, not that we're making anything better for them, but some of these plants may have actually been just naturally dwindling in certain areas where the climate was naturally warming as opposed to being accelerated by human impacts. So they may have been kind of dwindling on their own, and we're just making that faster, in other words. So this is kind of the habitat you find them in. These are the ravines here where there's usually a little drainage at the bottom that just cuts through and carves these wonderful features that harbor a lot of plants here. This down in here, it tends to be cooler and more moist, and you get a lot of things that just barely creep into Texas that you'd otherwise normally find like up in the Appalachians or the Ozarks that find refuge in these little spots here that just very locally provide the conditions that these plants want. There are certain plants that we'll look for when we're looking for Superpedium habitat. They almost always seem to be where you find this broad beech fern growing. This fern is another one of those species that is very specific to where it wants to grow in the proper soil chemistry and site conditions. And so this is oftentimes one of the more conspicuous things you can kind of look for if you're looking for Superpediums in the wild. Another thing is the wild comfrey. This will oftentimes too be growing with the lady slippers. It's another pretty specific, site-specific plant as well. So usually where you find slippers, you find this as well. So here's kind of an example of some perfect habitat. This is, it's hard to kind of see the topography here, but this is on a slope. It's got covered with the broad beech fern here. You got a comfrey in the middle there. And this is actually a site where there used to be a Superpedium, and it just disappeared in the past decade or so, according to, I never saw it, but this is according to Peter Luce. And again, flowering dogwoods, whether or not there seems to be a connection between these. I mean, dogwoods are around in East Texas, but they're not common enough where we would have that connection with practically every orchid is within the root zone of a flowering dogwood. And we're seeing the same pattern throughout the rest of the species range, not just with this species, with other species of native lady slippers too. So, and as you all might know, there are some disease impacts on Cornus Florida as well. So they're not entirely stable at the moment either, especially a lot of the southern coastal plain populations. So again, intervention is the solution. And so especially if we want to just at least preserve our Texas genetics. I mean, even though it may survive in other areas like up in the Ozarks or the Ouachitas or some of these other higher elevation cool areas, that doesn't mean we shouldn't just let our Texas populations just naturally go away. We should really preserve these genetics so that we can learn from them and just get them backed up. Both for maybe reintroduction as well as just ex situ conservation, getting these things preserved in other botanical gardens around the country. And so basically, yeah, we started in 2020. This was kind of something where Jared Barnes at Stephen F. Austin University, he was talking to Peter Zale at Longwood Gardens, who's kind of a wizard with native orchid conservation. They have a wonderful laboratory there for doing all the in vitro seed germination. If you're familiar with orchids, they're not something you can just necessarily throw seeds in the pot and just have them germinate like most traditional seeds. They do need either that fungal partner to be able to germinate, or they need some artificial conditions that basically will allow them to germinate in these controlled laboratory conditions. So Jared was talking to Peter, and I was independently talking to Peter too. And Jared and I didn't know that we were both talking about the same ideas here. And so basically, we once we kind of Peter brought us all together. I mean, I knew Jared, but didn't know he was working towards that. We all decided to just get together and go pollinate some orchids in 2020. And so that year, Jared brought some students out in the field, and we got 2 sites pollinated. We swapped pollen between 2 sites, and we collected seeds and got them going. So those are Jared's hands there pollinating this Superpedium here at one of the sites up near the town of San Augustine in East Texas. But after that, I basically wanted to continue doing that. Again, we only really got 2 sites done. We got some seeds out of those, but to do this right, we just really need to capture as much of the genetic diversity as possible. And so basically, I'm insane. I just decided I was going to get swap pollen between all these plants here. And so that's kind of what I did with the help of others. And so basically on April 4th, usually they start blooming around the first week of April. I drove the 200 or so miles to one of the populations. You can see my house is just south of College Station in the Milliken area. And went to the first site, which was Indian Mounds Recreation Area in Sabine National Forest. On the way, you get to see all sorts of cool stuff. So yeah, I mean, a lot of people think I'm nuts for driving just 2 hours one way just for a day trip to just go see plants, but you can see all these wonderful things on the way. The Baptisia, Sphaerocarpa, and the phlox were all flowering, and some of the nice little prairie remnants along the way, and the Pedicularis, woodlouse. I always try to bring my travel partner, Bungie, with me. He usually sleeps most of the way, but there, met up with Peter Luce. If you don't know Peter, he's just basically the person he's basically the brain trust of all the East Texas plants. He knows where every single rare plant is. And so he is just an invaluable resource for everything. He was one who I'm not originally from Texas. I came from Florida several years ago 2016, and he was basically the one who initiated me into where everything is. And so, and now as between he and I and the rest of our network of plant nerds that all get together and go out and find things, I mean, we're still always finding more stuff. It's not like everything has been found already, but yeah, Peter is the guy for as far as all the knowledge for this sort of thing. So we got to the first site, and the plants were, we had seen them the previous year on April 4th, all wide open, but this year they were still just almost there, but still a little bit droopy. I decided to collect some pollen from them anyway. I did not pollinate them, but I just went ahead and this one open, the most open one here, at least, I reached in and grabbed some pollen and put it in a little tube here, and got it on ice just to keep it cool, and figured I would take it to the next site, hoping that maybe the next site would have some flowers open. So I like snakes, and we get to see all sorts of beautiful wildlife along the way. And so we went to this other site here, and we were hoping maybe this would bloom this year, but it came back really pathetically. It was just it's what you're seeing there in the grass there, in the center of the image there. It was just a tiny little shoot, and so that was kind of disappointing that because this plant had been a much larger, bulkier plant the previous year. It hadn't flowered the previous year, so we were hoping it was building up energy to flower it, but it came back just so tiny for whatever reason. So that one was off the table this year. So I went to the next site, and we're glad to see that there were flowers there that were fully open and ready to go. So we basked in their glory there for a while there, with the light hitting them just right. I mean, it just never gets old when you see these things in the understory, and the light beams of light penetrate down and get them all backlit, and you can see all the details and all the little parts there. You can kind of get a sense there of the habitat in the ravine, growing on these slopes here, not far above the water. And there's Bungie enjoying it. So basically, when we're looking to pollinate these things, you got to be familiar with their anatomy here. And so basically, we got the anther where the what's labeled as the pollinium. Pollinium is basically in the orchids and a few other plants too. It's basically like these bundles of pollen. It's like a sticky mass of pollen that just kind of is meant to, basically that whole thing is meant to hitch a ride on an insect. It sticks to an insect, and it's just a big wad that's meant to be transferred to the stigma of another plant. And so basically you can see where the stigma is there, kind of offsetting from the anthers there. And so basically the way these plants are set up here is generally bees are the main pollinators, and so bees will be attracted to the flower through its scent, and will basically go into that pouch there, in that big gaping opening, and they'll be in that big pouch there. And basically the only way to get up is through the back, through the right side of that pouch there. There are some hairs that allow the plant to climb up. Anywhere else, it's just too slippery for the bee to climb, and the lip of that pouch is kind of curled in to also just help contain it. So basically the beetle or the bee is forced up the back and out where the arrow is, where it says bee exits. And in the process of doing so, it'll come in contact with the stigma and the anthers. As it's climbing up that back wall, clinging to those hairs, its back will basically rub up against the stigma first. And so if that bee is carrying the pollinia, the pollen bundle from a previous flower, it'll transfer that onto that stigma. And then as it's leaving again, one of those pollinia may end up sticking to the back of that bee to go carry it to another flower. These flowers offer no nectar to the bees, so it's basically they're just tricked to come there expecting something, and they never do. But obviously they go to another flower hoping for something there, even though they're not going to get it. but in the process, they do indeed get these plants pollinated. And so there's a kind of a close-up of the pollinia, or a pair of pollinia from one of the anthers of one of these orchids on the stuck to my forceps there. And it is literally stuck there. They are very sticky. They're meant to basically stick to these insect backs here. And so basically on another plant, we'll just take it and we'll stick it onto that stigma, which is what I'm doing there. And basically that's what it looks like. It'll just be this big clump, just literally just stuck there, and all that pollen will absorb into there, and everything will take place that needs to for fruit production. So basically from all these different sites, we'll be taking pollen and then collecting pollen, and basically nobody will be self-pollinated. Everybody will get pollen from somebody else, and we're keeping records of who got pollinated by who. And if one plant has several flowers, each of those flowers may get pollen from a different site. Again, just going for as much genetic diversity as possible. So it's a great time to be out in the field because you get to see all these other wonderful things in the ravines, all the trilliums. My dog likes smelling trilliums, I guess. We went to this other site, found yet another non-flowering one, hoping this one will flower this year. But that's just how it works sometimes. You can't have every single one flowering. And then got to this other site where we were really hopeful they would be flowering. This one tends to be always later than the other ones, and sure enough, it was. So we're going to have to wait on that one there. There was another little side ravine at that site with another one plant that we had got a seed from the previous years with Jared, but some animal had nipped the developing inflorescence off. So that one was off the table too. But growing right nearby it, we got to see some of the Uvularia perfoliata, a pretty rare plant in Texas, which grows in that one particular ravine there. So our last site we skipped because this Pat Roon site, because this one is always much later than the other ones. And so it was already pretty much a long day anyway, running all over between these different sites and hiking back, collecting pollen, bringing it back and getting it on ice. And so basically aside from what got pollinated, I had pollen basically that was going to be taken home and storing in the fridge for the next round of plants. Oh yeah, we have another one here that was just way too small. This was one that actually lost its dogwood and has just been really small and pathetic ever since then. So again, it just may show that relationship that it has there. But yeah, in the area we got to see some of the native azaleas flowering at that time too, and we were in the area, so we checked out this other orchid here, Isotria Verticillata, which is there's only like 2 sites left of this in Texas, and it's just such an alien orchid with that interesting whirl of leaves, and it's just very unusual. Just such a neat thing to see. There's Peter Luce for scale. He's photographing one over there while we've got some in the foreground here. Just kind of give you an estimate how big they are. I did pollinate those, but yeah, basically that whole day I traveled, and literally it was all one day. We between my house and going out there, running around between sites and coming all the way back, I put in 598 miles. Fortunately, Peter lives much closer. He lives up just a little bit east of Nacogdoches, so it's not that far for him. So next trip, we're ducking in the woods and got to be greeted by a nice, pretty cottonmouth, and also got to see some of the one of the last remaining sites of Heuchera americana, which this would be a good example of those things that most people would just look at that and say, well, who cares about that? It's just a pretty boring little plant. It doesn't have pretty flowers or anything, but it's a pretty rare plant in Texas. And it is of conservation concern in my mind if its population has been whittled down to maybe this one site. And then nearby, we found yet another Superpedium we were looking for that actually several of us were looking all over the site for this plant. They'd known where it was from previous years, and we had basically, I think it was 4 of us had been walking in circles around this plant forever. It's just it's hard to really understand how well these plants just blend into the environment among all the other common plants there, the smilax, like you can see there on the left, and all these other things. We basically gave up looking for it, and then on our way out, Tom Phillips, who's with the U.S. Forest Service, he's our botanist. He was the one who just suddenly saw it, not even looking for it after we had basically given up looking for it. But in the end, it was too small, so we had to pass that up. So we returned to that one of those sites where the flowers were almost open the previous trip, and one of the flowers at least was open. So we got that one pollinated, and went to that other site that we thought we were too early on, the previous site we didn't even look at, hoping that we might see something, but again, they were still a little bit too droopy. But after I was poking around at some and kind of lifted up the little hood there and was looking in, it was looking a little bit more more open by then, mostly due to my manual manipulation. So I just went ahead and took some pollen. It readily came out, so I figured it was ready. And we applied some pollen from one of the other sites, but left those other droopy flowers for next time. And just down the creek, there's also this other little patch of them that grows right along in a flood zone of this creek. And they had actually just been kind of buried under all this flood debris. We had actually kind of uncover these. It's kind of an unusual situation for this orchid. You normally don't see them in these kind of floodplain regions where they are subject to flooding, but obviously this one's been putting up with it. It's never bloomed that anybody has seen in recent times, but hopefully we'll get some blooms out of it eventually. So this next trip, we returned to the Matlock Hills site again, looking for those other 2 flowers to open, and they were getting there, but decided I knew I was going to be coming back to show some other folks the site, so I just held off with pollinating those, waiting to another time. And I had to be up in Dallas, so we stopped at Dogwood Canyon Audubon Preserve up there, a really amazing place with some of the westernmost disjunct flowering dogwoods. Again, that kind of host for the possible host, mycorrhizal host for the lady slipper orchids. Not that they're going to be up here, but it was just interesting to kind of be out of that typical East Texas piney woods ravine habitat and be seeing them up in this very different environment. Just kind of going to show how adaptable this species is. So after everything else I needed to do in Dallas, I basically started heading east again. Again, all on the same trip, driving around the state. So I figured I'd check that Patroon site again where they were almost open. But on the way, of course, I can't just go from point A to point B without looking at plants in between. There's another orchid that we've been working on, Calopogon oklahomensis, the Oklahoma grass pink orchid, that I was wanting to get seeds of. It's a little roadside population that's Mark Tietz found up in just west of Athens, Texas. And so they were still in bud, unfortunately, but this is what they should look like when they're flowering. So kind of missed it on those. And by the time I got back into East Texas to that Patroon site, it was dark and pouring rain. And I didn't care at that point. I mean, it's just, I don't mind the rain and don't mind the darkness. And so I've got a headlamp, but I don't know, I challenged myself. These are about a quarter mile off the road, no trail to them or anything. And I knew where to park, but without my landmarks, I just decided to walk in the dark, just using the lightning to illuminate my way and see if I could find them. I mean, if not, I'd just give up and use my headlamp. But it was just a really beautiful time to be out in the woods in the thunderstorm with lightning flashes, just kind of giving you temporary views of all the wonderful understory in there. And eventually I found them. I guess I'd been there so many times, I just knew right where to go. I flipped on my headlamp and there they were. But after a while, I mean, I was just totally drenched and just totally enjoying it. And I turned off my headlamp, and every time the lightning would flash, I was trying to capture what I was seeing. It was just such a wonderful thing where these orchids would just be illuminated. So this photo here is without flash or anything. I kept snapping my phone camera, just trying to catch it right at that proper moment when the lightning struck. And so basically all the illumination here is from a lightning flash here. Almost looks like a watercolor painting or something, but it was almost kind of as magical as just what I was seeing there. So this is just kind of why I do this stuff. It's just these wonderful experiences where you can escape reality and just be at one with such wonderful experiences, not just seeing them out on some sunny day. And so, yeah, that previous day I was going to be that evening I was rushing because I needed to get all the way back to Lufkin to meet up with Michael Eason, who you may know, who's obviously one of our Texas native plant geeks here. He's out in West Texas, but he travels all around the state, wrote The Wildflowers of Texas, and his girlfriend Marjeska came with us. And Romer, you can see Romer sticking his head out there. He's always we've all got dogs that come along with us on our travels. But yeah, that previous night I had to rush all the way back to the brewery, Angelina Brewery in Lufkin, to make sure we could all have beers together, which I finally made it, all drenched and everything. But we all went out the next morning, and it was just because I was still in East Texas. It was a Superpedium free day for a change. It's kind of exhausting, keep revisiting these same sites over and over again, even though it's wonderful to be able to be in their presence. But we did see some other orchids, including the Pogonia ophioglossoides and Malaxis unifolia, kind of a more cryptic orchid with little tiny green flowers. And then the day after that, I was still in East Texas, went back to those 2 remaining plants that still needed to be open, and they were finally fully open. And so they all got pollinated. And went back to those Isotria Verticillata to check on them, and was very happy to see that they were actually producing fruits here. You can see those kind of elongated things. You can see the remnants of the flower down there in the lower portion where with the ovary already just starting to swell, and then the top one there is already a little bit ahead of it. This population, Peter Luce had been watching it and had never noticed any fruits actually developing on these from natural pollination. So this was a first, and Peter Zale at Longwood Gardens was excited to try this just because nobody's been able to do this to germinate these artificially. And so he was trying to basically crack the code on these, and he had some ideas on how to do it. And and as an update on this, he finally got germination on these Texas plants here. I mean, this is a pretty wide-ranging species. It's not common anywhere, but it was our Texas plants that amounted to the first time this has ever been germinated using his various protocols he's been going through a lot of trial and error on. So we will get those genetics backed up. So by that point, all the flowers that we knew of, and at least there were a couple other sites I could have gone to, but this was just plenty for me to deal with. So basically we're waiting now 55 days. And in the meantime, I went back to that Calopogon ochlomensis site where they were still in bud before, all the way up near Athens, and they were in full flower. So we got them pollinated. On the right-hand side, you can see one of my very high-tech tools, which was a little piece of wire that I found alongside the road, which I used to kind of tease some pollen out of one plant and slap it on another one. You really don't need anything fancy for this. Sometimes I'll just grab a little piece of dried grass or a pine needle or something. It'll work just fine. You don't need anything fancy. But So yeah, we got those pollinated, and we've already got seedlings of this going. Spiranthes brevilabrus, this was another one that several of us hit up in Sam Houston National Forest, up just east of Huntsville. There's only a couple sites of this left, one on private land, and then two little populations in Walker County, and then a couple down in Galveston County on some private land. And that's basically it for Texas nowadays. So you can see all the records, they're shaded in the county records. Most of those don't exist anymore. And it's definitely more prevalent in Florida, where I became first familiar with it. But it's a prairie species, and it kind of grows kind of in this transitional geology. If you look in the upper left-hand photo, basically they're growing in that one little area where the vegetation changes away from the blue stem grass into that shorter grass there, and that's because the geology changes right there. And that's where the orchids are, where all those flags are. Those are marking individual plants that we're pollinating, and they're really tiny. And anywhere beyond there, up in that expansive bluegrass, they just don't like it up there, just because it's the wrong soil. And so these things, we were able to pollinate them and then get seeds out of them in basically 2 weeks, because that's how quickly they go from flowering to shedding their seed. And you can see the seed on the lower right-hand side there. It just basically looks like dust. That's basically the contents of that one capsule sitting on the envelope there. So 55 days later, May 31st, we went to check for ripe capsules on the orchids, and we were pleased to see that the first site we stopped at Indian Mounds, they were nice plump capsules waiting for us. And so naturally, these would actually, through the summer, they would actually dry, and it wouldn't be till fall that these would actually turn brown and basically crack open and dump their seeds. But for the purposes of growing them in vitro, you could collect seeds in, but you'd need to go through a lot of more complicated protocols to kind of overcome their seed dormancy and scarification, all sorts of other treatments that they would need at that point. Peter Zale at Longwood, he's kind of figured out a way where basically if you collect them while the pods are still green, the seeds will germinate like almost instantly once they're put on the suitable media in a petri dish or in a test tube. And so, and he's also found that they amount to much stronger plants too. When you do it that way, they'll grow much faster. And so that's why we're kind of unnaturally collecting them earlier than they would naturally be shedding seed. And so every flower we pollinated was successful. Some of these were on national forest land, and so through our permitting, we weren't allowed to collect every single seed. They did want to leave some of the pods on site for just natural dispersal and hope that there would be some natural seedling recruitment. Again, we don't normally see pods actually naturally forming here, so this was kind of like hoping this would actually help things, and we'll see some Superpediums popping up from these. I mean, the chances that one of these seeds is going to germinate is extremely low in the environment, but it's better than nothing here. So we basically collected only pods from the most diverse pollinations, just to get as much genetics as we could. And these all just get overnighted to Peter Zale at Longwood Gardens I've been talking about. Again, they have just a wonderful program there. They have they're very well funded, have unlimited resources, it seems, and basically he's just told me send them whatever and they'll get it going and get it backed up and get it shared around with other gardens and work with folks for getting things rewilded as well. And so basically this is what happens. I mean, the seeds are put on this kind of media, this kind of mixture of different nutrients and things. It does not have, sometimes when people do this type of growth, they will include a fungal culture as well. But for these purposes, these are totally what's called abiotic. They're not the seeds are not set up with the fungal symbiont that they need. They'll have everything they need with these nutrients here in these controlled conditions. Those little discs on the right are just little chunks of russet potatoes that kind of leach carbohydrates into the media that the seedlings need. but yeah, they just look like little worms or noodles or something at this stage. They're just basically um, that's basically what a little embryonic seedling Superpedium looks like at this stage, and then they look like some parasite or something that you hope wouldn't come out of your dog or whatever at this stage. And then after a while, they'll start looking like after several different treatments, they'll look like start looking like normal plants with leaves and things. So this is 20 months from basically when the seeds are germinated. This is a different species here, just for reference, but they all look the same when they're at this stage. And so, yeah, basically, once they're grown up to a suitable size, the plans are to replant at a number of suitable protected sites in East Texas, and also to back these up again in ex situ conservation, getting these in conservation collections with gardens both in Texas and around the country that have the proper conditions to be able to care for these. It's basically something that a lot of botanical gardens do in terms of ex situ conservation. Sometimes it is good to get these things away from where they're naturally imperiled, especially if there's like disease pressures or just suitable habitat is just dwindling, or it's going back to, again, if it's a climate where maybe they were not very happy and just kind of persisting, and they would be happier either further north or in cooler conditions. So basically I calculated it out, and through this whole trip going back and forth, pollinating all these different plants and revisiting for seed and stuff, I basically traveled 2,649 miles, which the width of the United States is 2,800 miles. So all that just kind of circulating around East Texas. And so now, basically in a few weeks here, we're going to be getting this bag of seedlings here. These are basically dormant seedlings. You can, if you look closely, you can see basically the buds where this season's leaves are going to be coming out. It's mostly roots though, but these are basically from those pollinations that we did with Jared Barnes and his students in 2020. And some of these are going to be replanted in the wild. Other ones, Haley Dunbar, who's a student at Stephen F. Austin State University under Jared Barnes, is going to be doing a study looking at different types of potting media and nutrition to be able to better learn how to maintain these in cultivation, again, for ex situ conservation, and also just for being able to hold them until they're up to a proper size for planting at these sites that we've identified in the wild. So she's going to take that on as one of her student projects there. So we're all looking forward to working with her. We'll be potting these up here in another couple of weeks and growing them out. Yeah, that's basically one of the many things I do in my insane life. I think I mentioned I didn't get paid for any of this, even though everybody thinks people are just throwing money at me to be traveling around doing plant conservation work and other things. I do definitely get paid through other means, but oftentimes I'm just putting it back into feeding my addiction here, and I don't know, it's just my nature, and a lot of people on the list here that have helped me out, and I'm probably forgetting some people here. It's just kind of their nature too. We're always just kind of out there asking ourselves, why do we do this? And I mean, we know why we do it, but in the end, it's basically a labor of love, and it's a passion, and really a lot of unsung heroes out there doing the same thing. So that's pretty much it. And if you like lots of plant photos, definitely follow me on social media. You'll that's another pathology of mine too, is I post way too much stuff from all my travels all over the place. So there's a lot of cool photos of not just plants, but geology and snakes and reptiles and all sorts of stuff. But yeah, I appreciate y'all having me, and thank you very much.
Wow.
Thank you for a wonderful presentation.
Yes, indeed.
Say, Adam, the first question that we have is about dogwood. So it's about the companion plant for these East Texas dogwoods. People want to know if the dogwoods that you find in the Dallas Dogwood Canyon can grow, are the ones that are able to grow in Austin, and having to do, I guess, with soil pH.
Yeah, well, that's the thing is, I know I'd given a talk recently to one of the chapters up in the Dallas area. Basically, I think people see that there are those dogwoods growing in Dogwood Canyon there in the Audubon Preserve, but you got to think that they're only growing where they want to grow. It's not like you can just take those out and just grow them anywhere in the Dallas-Fort Worth area. They're going to want that, just like the lady slipper orchids are growing in that just that little sweet spot to where the geology is right, the moisture levels are right in the soil, the chemistry, everything, the exposure. They're not just growing everywhere, so you're going to have to if you want to cultivate them even in the Dallas area from that particular site, you're going to need to be paying attention to the site conditions and make sure that you can give them what they provide. A lot of people fail with dogwoods just because they just think that, well, they grow in Texas, therefore they should grow in my yard, but that's not the case. Plants oftentimes really need very specific conditions, and I used to work in forest pathology at the University of Florida, and even there people would have problems with dogwoods. I mean, they are disease prone when they are stressed, and almost all the time nobody is planting them where they really want to be. They're just, their limits are just always being pushed, and everybody blames anthracnose and all the other diseases that are out there. But again, those are stress induced from being cultivated in the wrong spots. So yeah, I've never thought that dogwoods are really a good plant to be in typical just landscapes, or whether it be commercial or private. You can definitely go out of your way to try to create the right conditions for them, but it's not something most people are going to be doing. So that's kind of the long answer there.
Okay. Well, then also though, the East Texas dogwoods are very different than the ones that can grow in Central Texas. The roughleaf dogwood is what we get here in Fort Worth, and I think in Austin as well.
Right.
So
Yeah.
Go ahead.
Well, I was going to say there are some scattered populations all the way out to the Austin area that are definitely more tolerant of adverse conditions. That, like if you take a dogwood from a ravine in East Texas and you collect one from like one of the populations near Bastrop or out near the Atlanta River, you're probably going to, and you plant them like in the Austin area, you're probably going to have obviously better luck with some of those Western Provenance ones that tend to be in the drier conditions and different soils. So yeah, you got to know where your plants are coming from too.
Plants are happy best where they evolved.
Right.
So a question about the orchids. When does this species typically flower, and is native orchid poaching a major issue throughout Texas?
Well, the lady slippers specifically, they flower around the first few weeks of April. But as far as the various other orchids, I mean, we have most months of the year, you can find at least one species of orchid flowering somewhere in the state. So it all really depends on the species. And poaching, yeah, definitely, it's a big issue. We definitely do not share these locations with people. People see me posting these photos or see my presentations, and of course want to go see these wonderful things. And yeah, I mean, it's something I hate being kind of like, I think people just think I'm coming off as being elitist or something when I just kind of nicely tell them no. But when you have too many people who know about these plants, I mean, even if they're not going to poach them or share the localities with someone who may share it with someone else who ends up poaching it, just the amount of site pressure from people visiting during that flowering season is enough to really change those local conditions and change maybe change that soil flora with the fungi that they depend on, and from people tromping it around and compacting the soil and what have you. that it's going to be detrimental to the plant. So we really try to limit the number of people that see these every year, even amongst ourselves, basically.
So conservation of the habitat is really important here in a big way. Meg wants to know what particular bee species, or is there any particular bee species that pollinates the plants?
It's I've never actually seen them in Texas, but apparently it's several species of specifically bumblebees that are pollinating these. So usually larger bees are the ones that are what these orchids are kind of have kind of evolved to have a partnership with, and tricking them to coming to their flowers and getting smeared with pollen and
Crawling up the backside. Someone asks, do the lady slippers collect moisture for the bees to get a drink?
Not really. I mean, they a little bit of rainwater may end up in there, but it's not like it tends to dry up. I mean, they have that little kind of hood that kind of covers the opening of the pouch to try to shield as much. It tries to actually exclude water from itself. So generally, it's not really made to be like it's not made to hold moisture. It would definitely rot the flower if it does hold moisture for too long.
Oh, okay, that makes sense. So those, okay. How large is the inflorescence typically?
Well, the inflorescence, the whole thing can be about maybe 25 inches tall, 30 inches tall, maybe at the most. Definitely over 2 feet, but not quite 3 feet. And the flowers themselves are with those lateral sepals hanging down. I mean, some of them are really long and they may be like 8 inches long or so hanging down. So the entire flower measuring from those little dangly lateral sepals to the top of the hood, they may be like 8, maybe 10 inches tall.
Oh, wow. How stunning.
Maybe about 6 inches wide.
Gosh, that's big. How do you mark the sites to find them again, especially just one plant in a forest?
Well, at this point, we all know exactly how to walk right straight to them. So,
Okay.
and we've got, they're all on GPS as well. And these sites are well known to the state folks, so they've got it in their database. And yeah, they're basically all these plants are very well known plants among the few of us here. So.
Okay, not a question that I see here, but I'm curious, when you post pictures, do you try to strip off then the GPS data out of that?
I do, yes.
Yeah. Okay, Anita wants to know, what are the current inter and intra population genetic profiles of these populations? And do you plan to compare genetic profiles before and after interpopulation cross-pollination?
Yeah, that's one of those unknowns. We really don't know how genetically diverse these different populations are. We're just hoping that through all this cross-pollination that we're doing whatever we can to basically yield some diversity. But as we know from other plants and other studies, just because there's multiple individuals doesn't necessarily mean that you have any genetic diversity at all. So yeah, it's kind of an unknown. We're just kind of hoping for the best. It would be wonderful if some student had the funding to take on a study like that to be able to look at that more. But I mean, it's one of those things where we look for we want to have genetic diversity, but it's also kind of been shown through other studies with other plants that even if you have low diversity, it's not necessarily a bad thing. There are lots of natural examples where things are prospering, and in fact, some of our most successful weeds are basically go back to just one clone, basically one introduction. And so it shows that they can be strong, even though there's kind of this assumption that you should have as much genetics as possible within a population. So yeah, it's one of those things that we really need more information on this, and practically, I mean, that applies to every plant of, especially of conservation concern.
So a potential research grant for graduate student in the future, y'all.
Oh, definitely.
Beverly wants to know, how long is the pollen viable, or do you preserve it somehow to have it when other site populations are blooming?
Well, I mean, it's kind of an unknown. We're learning as we go, but I mean, generally the plants are flowering within a 2-week period at most. And I've been able to keep pollen in my refrigerator, just in a test tube, for 2 weeks and have yielded perfectly good seeds. And the seedlings themselves have been just as vigorous as those that received fresh pollen. So we at least, it's at least able to survive for 2 weeks under refrigerated conditions without any issue.
So do you ever pick up hits from iNaturalist?
I don't iNaturalist things.
Okay.
I have a love-hate relationship with iNaturalist. I have known a number of researchers who have had their sites basically destroyed just because someone iNatted it. I've seen a lot of things poached due to things being iNatted, even if they're obscured. If you know a little bit about the plant's site preferences, you can basically track things down. So basically any rare plants, I just don't iNat it. I will still either do herbarium specimens or photographic records and make sure sites are documented. But yeah, I mean, at the same time too, it's a great tool for that can be used for documenting these things if they're obscured on these rare plants at least. And yeah, I just personally err on the side of not doing it, I guess. Even though, I mean, when I go to a new place where I'm totally unfamiliar with, like if I'm traveling out of state or something, I will use iNaturalist. If I've got a short period of time and there's a couple plants I want to see, I will, and I don't know where to look, I may get on there and just see where some records, even if they're obscured, or are. And I guess I know I'm not going to poach anything or do anything unethical, but again, you never know who is and who's going to be using it for bad rather than good, basically.
Right.
But you can do that with herbarium records too, which are available online. So
Goodness.
it's just one of those things you're never totally gonna get around.
There'll always be someone, a rotten apple.
Yeah.
So Meg wants to know, how can you tell when Spiranthes seeds are ready to collect?
Yeah, well, basically it depends on the species. They're either going to be ready. Some of the smaller species are basically right after they're pollinated, they're ready 2 weeks later. Other species, it'll be a month later. I've never seen any of the species. I think we've done all species in the state now, and none of them have taken longer than a month. In fact, some of the smaller species, being that they flower in succession from the bottom up, you'll have ripe seeds on the bottom where the top will still be flowering. So yeah, it just really depends. You need to really watch them starting at like 2 weeks, and otherwise you'll miss them. They'll open up and dump their seeds very quickly.
Okay. Did you touch on how expansive the habitat for lady slipper has been in the past?
Yeah, I mean, at least in Texas, I mean, they are known to have been more widespread, and there are accounts of on Easter, people going out in the ravines and digging up plants and putting them in a pot and putting them on there, using them for centerpieces for Easter dinner, even though those plants, of course, ended up dying. But
Yeah.
people would apparently do this year after year. It's just a tradition to go have this for Easter Sunday. So it's well known that they were more plants out there, but still, I mean, being that their site preferences are very limited, it's going to be only where that proper geology and proper site conditions are, which is, when it comes down to it, it is pretty limited within the state. So I would not have ever called them common at any particular point or widespread at all. They're still going to be only in those very localized sites.
Okay, Anita Tiller says, with you at Britt, how about using the GGI gardens resources for collecting genetic material?
Yeah, we've already done that.
Okay, very good. Are these orchids declining in other parts of their range? I think you might have answered that. What can we learn from the orchid biology in the heart of their range, perhaps in places where they are happiest and healthiest?
Well, I mean, I think someone really needs to prove the association between the dogwoods and the lady slippers via the mycorrhizae. I mean, it just seems the more we look, the more that pattern just kind of holds true, but it's never really been scientifically proven. So one of the things we were thinking about doing was, and it may be part of Haley's study, was we were going to be, when we replant some things, we may identify some native dogwoods at these sites to be able to plant around the root zones, and some we will not plant around the root zones, and be able to monitor those over time to be able to really understand what they need. I mean, it's one of those things, again, going back to my past with forest pathology, we don't, aside from all these other diseases that do affect dogwoods, you never know when the next disease is going to come into the country, the next exotic disease that's going to wipe out a species or severely hinder a species. And if something comes in and basically knocks back all the dogwoods, that's going to be knocking back all these other associated species as well. And not just with lady slippers, but most orchids do have these associations with certain types of trees. So it's one of those things a lot of people don't really think about. When you lose one species, you have this trickle down effect to all these other things where there's just total dependence there. So it's I mean, it gets even more complex than that too. I mean, there's all sorts of other interrelations that go unseen in the under the soil beyond the mycorrhizae. That it's just an expanding field that as our science progresses, we're able to look at it differently. And so, yeah, there's just so many different directions we can go with not just lady slippers, but just botany in general.
Another area for graduate research.
Oh, yeah.
Have you found any species that parasitizes these orchids?
True parasites, not that I can think of. Never really see much in the way of pest issues with them. And I mean, that's pretty much the case with most of these kind of spring plants where they're basically in leaf for a few months out of the year and then they go dormant. Generally, they don't have any real permanent or even temporary real biological impacts in terms of pests or things that try to take advantage of them in different ways, either parasitically or otherwise. They tend to just throw up a leaf and they'll basically just store lots of energy into that rhizome there underground, and then they'll just disappear by summertime, and they'll basically be dormant underground through most of the year. So there's kind of a narrow window for things to really take advantage of these plants here. So I think a lot of these either parasitic plants or other types of pests tend to gravitate more towards those evergreen things, or those things that will just be a little bit more longer lived, where they can have more opportunity to suck the life out of them, basically.
Yeah. Do you preserve pollen for long-term genetic storage?
I don't really preserve anything. I get it around to other folks. But I think with other species of orchids, it has been looked into. I'm not well versed enough to really know the details on that because I've never really needed to. Again, I'm usually just working in that narrow time frame when these things are flowering here with current season pollen. So I've never really had a need to preserve it to the following year or anything. So I know for other plants it definitely can be done, but for other plants it also can't be done, or at least nobody's cracked the code on it yet. It's very different for different types of plants. So and our science is always changing. So it's just one of those things. If there is a need to do that, we would need to work out the details on it on a plant by plant basis.
Okay, and another question about the pollinator species, the bumblebee that does that. Do you feel like the pollinator decline might be affecting the orchid species reproduction?
Oh, absolutely. But I mean, I think at this point now, even if the bees were still there, again, the main issue is that the individual plants are just so widely scattered. None of these sites here have multiple plants on them. Well, there is one site in Texas that does have multiple plants, but at least all the ones that I've been focused on here, these are all just individual plants and or blooming plants at least. And it's just not possible for a bee to traverse the distance between one plant to another. They're just so widely spaced, so they're just not going to be naturally pollinated in the first place via the bees anymore.
Okay, I need to ask another question here. She says perhaps NAM Orchid Society would fund the inter and intra populations research on genetic samples you collected for GGI gardens. It's very exciting. She's very excited about your work, Adam.
We all are, I think.
Yeah, it's wonderful. Thank you so much.
Thank you so much. Yeah.
Thank you.